A very warm welcome on behalf of Biocrates. My name is Carmen Ludwig Papst. I serve the company as Director of Business Development. Thank you for the invitation and for giving us the opportunity to present today. Today I will talk about metabolomics profiling of population-based studies cohorts. This includes targeted profiling kits, population-based studies, and I will talk about combining metabolomics with genomics. The company profile of Biocrates in a snapshot. Biocrates is a company for targeted metabolomic kit technology. We are based in Innsbruck in Austria, Europe, and have an office in the USA in Boston. Biocrates has more than 15 years of experience. Our customers are spread in diverse fields such as pharma industry, academia, and health institutions. Biocrates holds a global community of more than 100 core facilities, pharma companies, CROs, and other customers. The technology is fully integrated mass spectrometry based, and it is across all indication areas. We are a unique worldwide provider of metabolomic kits for quantitative analysis of metabolites. Let's talk about targeted profiling and the readiness of technology. Biocrates products are standardized and quality controlled. It is a reliable, robust, transferable, and reproducible technology. That means we deliver comparability between MS platforms, and there is a good standardization between labs. This slide shows our profiling kits with our flagship product MXP-Quant 500. We develop our kits accordingly to FDA EMEA guidelines for bioanalytical methods. That leads to analytical, robust 630 metabolites with proven biological relevance. And this could be even extended of 240 sums and ratios by the quantitative measurement and calculation of metabo indicator. The kits have a 96-well format by default. It is important to note that you can work with the same assay in discovery, validation, and clinical environment. The technology can be used in all matrices and species where you expect metabolites. The next chapter is about population-based studies. I will talk about the key requirements of population-based studies, the principles of fair data, and the relevance and the clinical impact of fair data and population-based studies. So what are the key requirements of population-based studies? The analysis of cohorts require a broad coverage and robustness to deliver sustainable data and results. So the objectives of these studies, it has to be comprehensive, it has to be reproducible, it has to be sustainable, accessible, and scalable. And the key requirements which fulfill this is extensive coverage of dozens of biological relevant pathways, a robust analytical performance, pooling longitudinal and independent data sets that allows data reanalysis in different contexts. The technology must be broadly established, and thousands of samples must be analyzed in a cost-efficient and timely manner, in some cases over multi-labs and locations and countries. How to achieve fair compliant profiling in metabolomics. The solution we propose bases on 4 key elements. Measure and analyze large sample volumes in your network with scientific and economic excellence. Connect your science with other population-based studies globally. Leverage metabolite coverage and quantification for your data with clinical relevance. Do all this in an environment where methods and data are standardized and quality controlled, quantifiable and reproducible, as well as fair. It's all about fairness, the relevance of connecting science, and building on your network. Our platform enables fair data principle. What is fair? What are the fair requirements? It is findable, accessible, interoperable, reusable. This is the definition, the international definition of fair requirements. And what is Biocrates' kit solution to achieve fair requirements? You have the total ownership of data, including access to raw data. Uploading to databases is by default supported. You have a harmonized and standardized data format in one combined database, and metabolomics data from different experiments can be easily pooled with lab-to-lab comparability. One more remark, there is a white paper published under the name of Fair Compliant Metabolomics Profiling of Population-Based Studies. Come back to me if you're interested here. Year over year, we have increased the coverage of our ready-to-use large panel profiling kit significantly, and now we report up to 864 quantitative data points. The kit is standardized and quality controlled and leads to a harmonized workflow and stores data in a harmonized format. The kit delivers quantitative and reproducible data, leading to comparability across instruments, labs, and users. The kit enables pooling of independent data sets. The concept enables worldwide to contribute to a scalable and integratable dataset, and this is proven by ring trials. We provide total ownership of the results within a global network based on fair methods, enabling data principles. How can Biocrates deliver fairness across laboratories and instruments? This is based on the kit components, which consist of reagents and consumables. They include internal standards, calibrators, and QC samples. Else, we deliver prepared methods and protocols. So sample prep and LC and MS method are ready to measure. The workflow manager guides you through the process, it's called MetIDQ, and reads out the data and creates an automated technical validation report. This strategy leads to the analytical robustness, but also to the comparability across multiple MS instruments. Finally, all data are stored in harmonized and standardized data format in a combined database, giving you total ownership of the results within a worldwide network. And this is based on fair methods, enabling the fair data principle. Biocrates kits are research kits only, but as mentioned, we develop our kits according to FDA EMA guidelines for bioanalytical methods. Up to 630 metabolites could be extended by up to 234 quantitative data points of sums and ratios by the software MetaboIndicator, which also is backed by scientific literature to study relevant pathways in addition to metabolites. This could give access underlying enzyme activity and gain clinical insights. For example, if you look into the middle of the slide, IDO activity is measured by the ratio of kynurenine and tryptophan. The products are applicable for diagnostic, since the development is under FDA guidelines. The achieved biomarkers and the biomarker signatures are, so to speak, pre-validated. We have a global footprint. The shown circles give an example of cohorts measured with our technology. This shows the robustness of the technology. And think of it, data of fantastic cohorts are waiting to be reused or reanalyzed in a different context or integrated in multiomics analysis. I summarized some notable cohorts measured with our technology. For neurodegenerative diseases, ADNI, the US cohort of the Barack Obama Foundation, should be mentioned, as well as the BLSA study. Examples for other well-known population-based studies in Europe are Fenland, CORA, or APIC. Many data do exist of all the other population-based studies. All of these data could be reused and integrated into future studies. The biggest study definitely is EPIC with 25,000 samples. Now I would like to proceed with a topic which gains attractivity from year to year, and is the fact that you can combine metabolomics with genomics, MGVAS. So we'll talk about MGVAS, GIMS, and MGVAS databases. MGVAS stands for Metabolite Genome-Wide Association Studies. If we take three layers into consideration, the genome, the metabolome, and the clinical phenotype, there is a strong association between the genome and the metabolome since they're in close proximity. The metabolome now may bridge the distance of the genome with the clinical phenotype, which are in weaker association. This situation strongly suggests combining genetic data and metabolomic data and study the functional relation towards the clinical phenotype. In the last years, scientists took evidence of this fact and started to combine genetic data with metabolomic data, since some genetic results do not reveal the pathways and function the SNP is involved in. So MGVAS could brighten the functional relation towards the clinical phenotype. By enabling pathways analysis of the genetic dataset and help to distinguish phenotype converting variations, GIMS, which are genetically influenced metabolites, and to distinguish it from GIMS without influence. This may be useful to answer questions about treatment and response and may be important for drug development. And we should not forget that MGVAS provides a great benefit with little additional efforts. To underline what I have told so far about GIM and MGVAS, I would like to share a publication which, by the way, was the very first MGVAS study ever, and it was part of the cohort study. This study is an example of a GIM, a genetically influenced metabotype, and here we see a SNP of a fatty acid desaturase that was strongly associated with the glycerolipid levels. These findings were only possible when adding metabolomics to genetic data. So metabolomics may provide the missing functional link between the previously observed associations of SNP and serum and LDL and HDL and total cholesterol levels. So GIM may influence the individual susceptibility for certain phenotypes. The importance of mGWAS studies is underlined by the fact that there are some very good databases existing which already link genetic data and metabolomic data. So it's the Metabolomics GWAS server, it's the single nucleotide polymorphism annotator, the SNPA, and it's the OmniScience. All these are very serious databases and they link diseases, they link pathways to certain genes. This leads me to my last slide and to summarize what we have heard today. About the technology, it is a ready-to-use kit in a 96-well format, reporting up to 864 fully quantitative data points in a harmonized data format. It is a reproducible technology. It delivers lab-to-lab comparability, and this is proved in diverse ring trials. And you have total ownership of your data, enabling fair data compliance. We've heard about population-based studies and cohort studies, proven track record in large epidemiological population studies. I talked about the clinical relevance and analytical robustness through metabo indicator and the ratios and the sums. And I opened the mind for getting access to multicentric scalable solutions for large epidemiological population-based studies or cohorts for up to 35,000 samples per year. So it's really high throughput. The solution is that future workflow management cohort data can be combined with other omics data, and mGWAS matters to establish the functional relation between a genetic variance and a disease. And this is because you can distinguish GIMs, which matter as a phenotype converting variation, and you can distinguish that from a genetic variation with minor effect size. Thank you for listening, and thank you for giving us the opportunity to talk today. If you would like to take the opportunity to get in contact, in Korea we are in strong cooperation with omics. Both email address, mine and omics, are listed here. And now I'm open to take questions. Thank you very much.